Hey Team Bio, welcome to your final screencast on cellular respiration. So just a quick reminder, I know this is really tiny, sorry Campbell. This is our flowchart in the upper left-hand corner of the document. We've talked about cellular respiration, how there are two forms, anaerobic, which happens in the absence of oxygen, and aerobic, which happens in the presence of oxygen, but both of them start with glycolysis. This produces 2 ATP, and also here some NADH, sorry, not NADPH. The whole point of fermentation is to, so I guess I should also include, this is, way too big. 2 NADH. The whole purpose of anaerobic cellular respiration fermentation is to recycle NADH to NAD+, get rid of those high energy electrons that NADH is carrying. However, if we have oxygen present, then we can use these high energy electrons to do some important cellular work and make ATP. And this next piece, the aerobic pathway, I'm going to be talking to you about how this is actually made. Because if you look here, you see that glycolysis makes 2 ATP, and alcoholic fermentation doesn't make any ATP at all. It just recycles NADH to NAD+. So you're only getting 2 ATP per molecule of glucose if you're respirating anaerobically. However, if you're respirating aerobically, you are making somewhere between 36 to 38 ATP. Yes, so let's see. Okay, so glycolysis. Boop, 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 boop. We talked about this in great detail. You know that glucose comes in, pyruvate comes out. We spend 2 ATP, we make 4, so on net we get 2 and 2 NADH. As I said before, the anaerobic pathway just recycles NADH back to NAD+, so we can keep running glycolysis. And in the presence of oxygen, then we can more completely combust this glucose molecule. So we move from the cytoplasm where glycolysis is happening. Remember, this is happening in the cytoplasm. For the rest of aerobic cellular respiration, we are going to move into the mitochondria. We first begin with a reaction called the prep steps. So remember, glucose is a 6-carbon molecule. We split it in half to form 2 3-carbon molecules that were pyruvate. So for every 1 molecule of glucose, 2 pyruvate molecules enter the mitochondria. So here, let's just make this, correct our stoichiometry here. So we have 2 pyruvate, and pyruvate binds with a special coenzyme. It's called coenzyme A. And in this process, it's going to perform a redox reaction, and 2 high-energy electrons are going to be stripped off of pyruvate. Actually, 4 high-energy electrons in total. So NAD+ is going to pick up 2 high-energy electrons to become NADH. But this is going to happen twice, remember, because there are 2 molecules of pyruvate entering for 1 molecule of glucose. And out is going to pop a compound called acetyl-CoA. So this is a pyruvate that's been chopped to now become a 2-carbon compound and joining with coenzyme A. So acetyl-CoA is our carbon compound. This reaction is going to produce some CO2, which if you are a person, this will eventually reach your lungs and be exhaled. And this is also going to produce some NADH, which remember is carrying high energy electrons for us. Now, These acetyl-CoA that are, oh, I forgot this is a 2, and there should be a 2 here as well. So 2 acetyl-CoA are created, 2 CO2 are created. Why did that all erase? 2 NADH are created. Okay, so acetyl-CoA is going to enter what's called the Krebs cycle, or also called the citric acid cycle. You can use these terms interchangeably. Krebs is a person, and then the citric acid cycle is named for the first compound that's created after acetyl-CoA enters the process. So this is going to look very similar to the light-independent reactions. It's a cycle that is contingent on the input of a product to keep spinning. So in this case, the product, or the input, it's not a product, I guess it's a product of the prep steps, and it's a reactant in the Krebs cycle. So the reactant in the Krebs cycle is acetyl-CoA. This organic compound, acetyl-CoA, joins a 4-carbon molecule called oxaloacetate. You've actually already seen this 4-carbon compound before when we talked about the C4 pathway. This is how plants are storing carbon dioxide, the plants that live in dry or arid conditions. So acetyl-CoA, 2-carbon compound. Remember, I'm just drawing the carbons here. We're not going to worry about all the different functional groups hanging off of these things. But know that it's a little bit more complicated than just carbon here. But this 2-carbon compound joins this 4-carbon compound, and it makes a 6-carbon compound called citric, or citric acid. It is a slightly acidic compound, which is why it is called citric acid. Okay, we are going to take this citric acid and we are going to slowly combust it, meaning we're going to strip high energy electrons from it in a series of reduction oxidation reactions. So remember, we were talking about redox reactions before break, and if you lose electrons, you are oxidized, and if you gain electrons, you are reduced. So whenever electrons move around from one compound to another, something is oxidized, something is reduced. A helpful acronym is LEO, loss of electrons, oxidation, goes GER, gain of electrons, reduction. Okay, the LEO line goes GER. Okay, so we're going to slowly combust citrate. We're going to strip high energy electrons from it and send them to a process called electron transport phosphorylation. So here's what happens. Citrate is the first thing that is going to be oxidized. We are going to strip electrons from it and put them on our favorite high energy electron carrier, and that is NAD+. I don't know why my pencil is going so crazy. Okay, so NAD+ is going to swoop in here and grab high energy electrons, and it's going to become NADH in the process. This NADH is going to bring the high energy electrons and drop them off, oh my gosh, and drop them off at electron transport phosphorylation later, and in that process it's going to be recycled back into NAD+. So this is an example of a redox reaction where citrate is oxidized and NADH is reduced. In the process, there is also a molecule, or a molecule of CO2 is broken off of citrate. And this will work its way, if it's in a human, to the lungs where it's going to be exhaled. So then from citrate, we create another a 5-carbon compound called alpha-ketoglutarate. Now, it's not important that you know these names. I don't expect you to know or remember them. I just want you to know that electrons are stripped, a CO2 breaks off, and we're going to use these electrons later in electron transport phosphorylation. So the same thing happens to alpha-ketoglutarate. NAD plus swoops in here, it picks up electrons, it becomes NADH, and it's going to drop those electrons off back at electron transport phosphorylation, which we'll get to in a second, and it's going to become NAD+. Once again, this is an example of a redox reaction, and in this process, a CO2 is broken off from alpha-ketoglutarate, and that CO2 will be exhaled eventually. Okay, from alpha-ketoglutarate, when we break it apart, we get a compound called succinyl-CoA. In this process, moving from succinyl-CoA to succinate, we can make an ATP from ADP and phosphate, inorganic phosphate. We perform what's called a substrate phosphorylation level reaction. So we make 1 ATP here. That process, stripping off some energy from this compound, makes it into succinate. And we have another high energy electron carrier. It's got a slightly different structure and a different name, but it's very similar to NAD+. So it's called FAD. FAD swoops in here, does a redox reaction, and becomes FADH2. And again, it's going to go drop off its electrons at electron transport phosphorylation. High energy electrons joining the ones that NAD drops off, and it's going to be recycled back into FADH. And just like before, this is a redox reaction. This reaction converts succinate into another 4-carbon compound called fumarate. Fumarate requires a little input of H2O to become another 4-carbon compound called malate. Malate is going to undergo another redox reaction where NAD+ swoops in here, picks up higher energy electrons, becomes NADH, and they will be dropped off at electron transport phosphorylation. And once again, this is a redox reaction. And that final process converts malate back to oxaloacetate. And then oxaloacetate is ready to bind to another acetyl-CoA, and the Krebs cycle is ready to spin again. So the Krebs cycle has to spin 2 times. To combust 1 molecule of glucose. So tracing this back, if 1 acetyl-CoA enters this cycle and we exhale 2 CO2, well, acetyl-CoA came from pyruvate. We had 2 pyruvates that were created from glucose. So in order to break down 2 acetyl-CoAs, we need to spin this once and then spin it again. So the Krebs cycle spins 2 times to combust 1 molecule of glucose. Okay, so when I keep talking about electron transport phosphorylation, what the heck am I talking about? Well, if you zoom in over to this side of your note, you'll notice something that we looked at in class today. This is the membrane of the mitochondria. This is called the intermembrane space right here, and this is the mitochondrial matrix. Here we have a graphic showing us what happens when NADH brings our high energy electrons, which are depicted here and here, to an electron transport chain. Well, as you know, electrons can move through an ETS, an electron transport system, and they can use their energy to pump hydrogen through a membrane. And that's exactly what happens here. So NADH drops off its electrons at the beginning part of the electron transport chain, and hydrogen can be pumped through the membrane in 3 places: here, here, and here. This creates a concentration gradient, and this concentration gradient drives the formation of our favorite molecule, ATP, when it passes from an area of high concentration through ATP synthase, spinning this enzyme and phosphorylating ADP and phosphate into ATP. We call this chemiosmosis. FAD drops off its electrons a little bit later in the chain and only pumps hydrogen through in 2 places. So you get a little bit more hydrogen, a little bit more concentration gradient built up when you use NADH instead of FADH2. You'll notice something important is happening at the very end of the chain. The electrons have to be picked up, and the thing that picks them up is oxygen. Oxygen combines with these now low energy electrons. Remember, they were high energy electrons here, but we used their energy to pump the hydrogen through the membrane. So when they get to the other side, they're now low energy electrons, and they're deposited on oxygen and then some hydrogen in solution, and they form H2O. So there's kind of a beautiful symmetry here with photosynthesis and cellular respiration. Photosynthesis starts with the photolysis of water. Remember, we had to break down water in order to extract its low energy electrons and then excite them with the photosystems. And then ultimately those high energy electrons in photosynthesis ended up on glucose. In cellular respiration, we're stripping those high energy electrons away from glucose. We're using them to do work. They finish the process as low energy electrons, and then we dump them back onto water. So yeah, that is cellular respiration. Is there anything else I want to tell you guys? I don't think so. Oh, yeah, I guess so. We should do some accounting here at the end. So glycolysis, in glycolysis, we made 2 ATP and 2 NADH. In the prep steps, we made 2 NADH, and in the Krebs cycle, we made 2 ATP, because remember, the Krebs cycle has to spin 2 times for every molecule of glucose. So 2 ATP, 6 NADH, and 2 FADH2. So NADH and FADH2 are both, they're all going to drop off their electrons at the electron transport chain, and this is how we're going to make a bunch of ATP. So that's all. Okay, see you in class tomorrow.